four-dimensional. We have three dimensions and
yes, the smell. It's like you are visiting the Disney World.
Oh.
Okay, thanks to Jack, I'm here to tell you what I do in my lab, the regulation of signal transduction pathways. Now, you might have noticed those students start gimmicking because of the smell. Could you, I see.
Yeah, here, I got it more.
All right, so what we do is very similar to their response to odorants, all right? So signal transduction pathways, how do cells sense and respond to signals? Now, I do not use human beings as the model organism. I'm using the budding yeast as the model organism, right? So basically, there's a signaling molecule, elicit response. We try to understand how the odorant is interpreted in human beings. That's using animal as a model. We use budding yeast as a model organism. Alright, this is our model organism, the budding yeast. These are scanning electron micrographs. When I did this when I was a college student in China, so it's pretty advanced. And the size of yeast cells is about 2 to 5 micrometer across. So they are microbes, but they are also eukaryotic. So this has a huge advantage of a prokaryotic system. Now, why do we use yeast as a model organism? Because they're easy to maintain and they're easy to grow. And most importantly, there's so-called awesome power of yeast genetics. Now, you have done some yeast genetics already. Last week, when you cross two red mutants together, you generate diploids, which are whites, right? So have you figured out why? Now, the important thing about yeast genetics is the availability of a large collection of tools for us to dissect responses like the way we human beings respond to odorants like that. Right. Now, what do? We study how cells sense and respond to changes in internal and external environment. And for this kind of studies, generally there's kind of two processes. The earlier stage is to identify proteins that function in a signaling pathway. Now, that genetic part you guys did last week, right? Now, once you identify components, and the next thing is you try to figure out how these proteins work together to mediate signal transduction. For example, when you smell the odorant put in front of you, you kind of just pull back and then give disgusting kind of expression, stuff like that. Now, think about what's the receptor for the odorant. So everything pretty much has a receptor in cells that elicit response, and then proteins will interpret the signal, and then you're going to have changes in the cellular response. Now, yeast cells, actually, one of the most important aspects of yeast research is the fact that yeast cells can exist in stable haploid stage and diploid stage. Now, you have worked with haploid mutants last week and Y type. Now, this is very, very important because yeast cells are haploid, so if you generate mutation, you're going to have phenotype because you're not going to have the Y type gene from the other chromosome that complement the mutation. So using haploid yeast strains, we can generate mutants in a specific pathway by looking for certain phenotypes. For example, red color phenotypes you guys studied, ADA1, ADA2, right? Two of them. They give the same phenotype, but we know they're different genes because when you cross them, they produce wild type phenotype, white. Right? Yeast cells, the stable haploid stage, and they can mate when given chances, and they may produce diploid. Now, diploid cells are also very stable, and then on the harsh conditions, diploid cells sporulate. Now, this is another aspect of yeast genetics that's very important. The fact that the meiosis, any meiotic event, will generate 4 spores, right? Or gametes in animals, right? In the case of yeast genetics, that in the brewing yeast, budding yeast, the 4 spores will be packaged in a little sac, okay? And we can take the little sac and then dissect it. Well, let's see if I have to. Okay. So this would be this would be a tetrad. Those 4 colonies are from the 4 spores that are packaged in a little sac. This is very, very powerful. This allows us to generate mutant strands and cross them, generate double, triple mutants as we like. Now, once we started working with haploid strains, and we have a pathway in mind where we have a phenotype looking for, and then we can generate mutants. Right? Now, supposedly we have several hundred mutants in mating type A strain, 700 mutants in mating type alpha strain. Now, it's not true that you have several hundred proteins that participate in a pathway. So you're going to place those mutations into so-called complementing groups. Like we have 2 different type mutants last week. You had 2, other 1, other 2. And we know they're different because when you cross other 1, other 2 together, you're going to see white colony. And it means that there are 2 different genes that when mutated leading to the red color phenotype. So complementing group analysis simplifies analysis of mutants. You can place hundreds of mutants into half a dozen, dozen of genetic groups, basically genes. Now, yeast genetics and Nobel Prizes. Yeast genetics, despite yeast cells are really small. And so far, there are 3 Nobel Prizes awarded. Last one was to Dr. Yasunori Asumi in autophagy, and vesicle trafficking by Rennie Sheckman in 2013, and cell cycle by Dr. Hartwell. Now, these scientists all started with working with the same thing. They generated mutants in haploid strain, and then they do crossings like you guys did last week, and placing mutants into so-called complementing groups. So Dr. Hartwell probably isolated several hundred mutants that are rested at different stages of cell division, cell cycle. All right. Now, and then he placed them into complementing groups, probably 20, 30 of them. And then vesicle trafficking secretion mutants, probably same number, and autophagy around 30. All right. So the power of yeast genetics relying on the ability to cross mutants, so you can play them into fewer number of complementing groups. All right, so you have done the red color phenotype, and just show you that this is a 2-to-2 segregation of two red colonies from either one mutation and 2 white type cells. Basically, this just show you one of the very important aspects of yeast genetics, tetrad analysis. Now, using the red color phenotypes, right, we can actually isolate mutations in a specific pathway. Now, this white type strain, I see this kind of red sectoring colonies. Using this type of analysis, right, we can identify unknown components of signaling pathway. Now, I don't want to bore you with details based on the color phenotype, and we can identify genes in a specific pathway. For example, this important kinase, TOR2 kinase, it's essential. It is a temperature-sensitive allele at 37 degrees. It's not viable. Now, TOR2 kinase is highly conserved from yeast to humans. By studying the function of yeast TOR2 kinase function, we can extrapolate the function in human cells as well. Now, using the genetic analysis like the way you did, and we identified SUC7 mutation, use the color phenotype, and SUC7 mutation, when mutated, it can suppress the mutant phenotype. As you see, TOR2 mutants now can survive now due to another mutation of SUC7 or 511. And then using genetic analysis, we can tell the SUC7 and 511 working different pathways to suppress TOR2 lethality due to TOR2 mutation. What else can we do with yeast lab? And we can start putting localizations. This is done by my ex-PhD student, and basically we're studying the complex. And it's not very clear here, but proteins localized to ER, endoplasmic reticulum. We can also use the SDS page and determine post-translational modifications. Now, when you identify proteins that work in a specific pathway, next question is how they work together to transduce a signal. like you responded to beta capital ethanol, like pullback, and then give disgusting look and expression. And so we started this by looking at protein localization, by looking at protein translational, post-translational modification. Now, currently, we focus on one project in the lab, the regulation of mitochondrial biogenesis. This work was done by Stephen Tell, who won Nobel Prize for this type of study 3 years ago, high-resolution microscopy. Basically, scientists now can overcome the limitation of light diffraction. Right. So as you can see, when cells grow in glucose, you have the basal level mitochondria indicated by green stuff here. And when cells switch from glucose to glycerol or lactate, and you can see there's tremendous increase in mitochondrial amount and volume. So we have isolated many genes, proteins, working in a switch from low amount of mitochondria to the high amount of mitochondria. Now we're using tools like protein localization, protein modification, and protein-protein actions. Try to dissect the mechanism behind the transition from low levels of mitochondria to high levels of mitochondria. Now, what can we learn from this? Well, the mechanisms by which a group of proteins work together to carry out a specific cellular process. In this case, the regulation of mitochondrial biogenesis. Now, the future outlook, what can we do with yeast genetics? This is actually the most exciting part of it. Yeast cells actually are the leading organisms that produce biofuels and pharmaceuticals. Apart from, we try to understand the fundamental cellular process, such as the regulation of mitochondrial biogenesis. All right, thank you for your attention. Boy, 15 minutes.